time in the morning. We just agreed not to do huge introductions because the speakers have far more important things to say than I do, but I'd like to introduce our first one straight away, Robert Lanza, the Chief Scientific Officer of Advanced Cell Technologies. He's making the running in a lot of translation of stem cell research into the clinic. Is a professor at Wake Forest, author of many books. I'm sure many of you are familiar with his work. And Robert, please, the stage is yours.
Thank you very much, and I'd like to thank the organizers for inviting me here today. So I will be speaking to you today about ES and iPS cells and moving into the clinic. And as you know, there are two major ways to create patient-specific cells. Of course, the first would be somatic cell nuclear transfer or cloning, SCNT, and the other, of course, is through cell reprogramming, generating iPS cells. Whether you use SCNT or iPS technology, this experiment we did a few years ago, it illustrates the power of these technologies. So these are some old cows equivalent of humans that are, say, 70 or 80 years old. What we did is we took a skin biopsy from the ear and we created hematopoietic stem cells. But because these are clone cells, we need to knock in a neomycin cassette so we can discern those transplanted cells from self. We then injected those cells into the respective nuclear donor animal. We didn't need any myelosuppression or myeloablation because the cells were very youthful, and through competitive repopulation, were able to take a home in these animals. And when we took some blood samples from these animals, we were able to show how the neomycin cassette started appearing in the first 12 days in the peripheral blood. By half a year later, when we looked at the colony-forming cells, about 60% of the cells were of clonal origin. And when we plated those out, we saw that they were able to generate giant white blood cell colonies, which you usually only see with very youthful cells. So again, the goal here is not just to give an old cow an immune system, but there are over 100 autoimmune diseases in humans with this type of technology that could be used. A similar experiment, this again is an experiment where we, in this case, it was the mouse model, severe myocardial infarct model, where we created cloned c-KIT positive cells, and those were injected at the margin of the infarct. This is the infarct, this is the healthy tissue. Again, this is a cross-section through the heart. And within 1 month, we were able to see the clone cells, which in this case were LAC-Z positive here, infiltrate through the damaged tissue and actually replace 38% of the myocardium. But until these patient-specific therapies are developed further and shown to be safe, the most likely first clinical translations are going to be in immune-privileged sites such as the brain and the eye. So at Advanced Cell Technology, we're focused in on the eye. We have a number of programs. We're currently in clinical trials using retinal pigment epithelium for macular degeneration, both AMD and Stargardt's. We also recently got FDA approval to use these cells to treat myopia. We also have a program where we're using retinal ganglion progenitors to treat glaucoma. We're also exploring corneal endothelium for corneal disease. We're also investigating mesenchymal stem cells for uveitis and glaucoma. And we also have a program using retinal neural progenitors for photoreceptor replacement as well as for neural protection. So, as I mentioned, we have now received FDA approval for 2 human clinical trials using the RPE to treat macular degeneration. The first is for dry AMD, and the other is an orphan indication, Stargardt's disease, which is the leading cause of juvenile blindness. AMD alone, as you may know, affects over 30 million people worldwide. It's the leading cause of blindness in patients over the age of 55 in the U.S. and in most developed countries. And the degeneration of the RPE cells are thought to play a critical role in the development of this disease. So we believe that stem cells are a potentially important source of RPE for transplantation into these patients. So this is just a cartoon of the eye. So as light enters into the eye, it hits the photoreceptors. These are the cones of the rods that we see with. And underneath those are the RPE cells. And these are the life support to the photoreceptors. So they provide critical nutrients, growth factors, they recycle vitamin A, they also phagocytose the shed photoreceptor fragments, a number of different functions that are critical for the maintenance of that photoreceptor environment. So, of course, there are many reasons why iPS and ES-derived tissues are ideal for regenerative medicine. You can create a virtually unlimited supply of cells. These are immortal cells that will grow in vitro, obviously, forever. They can be derived under GMP conditions, under pathogen-free. They can be produced with minimal batch-to-batch variation, and they can be thoroughly characterized to ensure optimal performance. And we have now generated RPE very reliably from both iPS and ES cells, almost with 100% efficiency. So this just shows you how you isolate these cells. So if you let your ES colonies to spontaneously differentiate, eventually all of those will eventually start to develop these pigmented patches, these little freckles in your dish. And those turn out to be the RPE cells. And they can be removed mechanically, and then you can plate those out. And what will happen is that you'll start to get outgrowths, and the cells will de-differentiate, lose their pigmented morphology, and then expand to confluence. And then once they reach confluence, they then reestablish their pigmented cobblestone morphology. And they'll go through this differentiation, de-differentiation phase over and over, so you can get very, very large numbers of these cells very easily. And we have extensively characterized these. You see a downregulation of the markers of pluripotency, and upregulation of the various RPE-specific markers. And also, one of the key functions here is that, again, responsible for phagocytosis in the shed photoreceptors. So you can test this by just throwing some latex beads in your culture dish, and you'll see that the cells eat them here, and you can actually quantitate that using flow cytometry. So they look like RPE and seem in every respect to be RPE. So we wanted to see what they could do in animals, in diseased animals. So we tested a couple of different models. The first was the RCS rat, the Royal College of Surgeons rat, and we were able to show that we could rescue the photoreceptor function and improve visual acuity in a dose-related fashion. Similarly, in the mouse Stargardt's model, we were able to prevent the onset of the disease. And if you look at these animals after the transplantation of these cells, you can see a pretty dramatic effect. So after about 100 days in the RCS rat here, there's only one layer of photoreceptors left. These animals are essentially blind. After the RPE injection, again, these are the human RPE that were transplanted to patients, you see very robust rescue of the outer nucleated layer that's about 5 to 7 layers thick. We have perfected this manufacturing process. Of course, the regulatory agencies are mainly concerned about the possibility of teratomers or your cells turning into unwanted cell types. So we've developed an assay where we can detect even one undifferentiated cell in over 1 million cells. In our particular case, the maximum clinical dose is only 200,000, so we can easily detect even a single cell, even though we know from in vivo spiking studies you need at least an order of magnitude 6 orders more to get a teratoma. Even if you spike in over 10% undifferentiated cells late in the procedure, you still don't end up with any undifferentiated cells. We've extensively characterized, I've studied these animals in safety studies. We haven't seen any safety signals, no tumors, no ectopic tissue. We've looked at long-term lifetime studies in these animals, and we haven't seen any teratomas. So based on that data, the FDA gave us the green light to proceed with clinical trials in the U.S. We have 4 sites in the U.S. The George Stein Eye Institute, Rose Eye, Jasper Palmer Eye Institute, and Mass Pioneer. We've treated 12 patients who have dry AMD. This is a dose escalation study, so we start out with 50, and then we move up to 100, 150, 200,000 cells. And similar design, Stargardt's trial here. We've treated 9 patients there, and we also have a study here in Europe at Huddersfield Eye Hospital, and so we've treated 9 Stargardt's patients. So early last year, we published a paper in the Lanza on the first 2 patients. The first Stargardt's patient, just for instance, only had hand motion. Within 1 week after injection of these RPE cells, the patient would start counting fingers, and then within 2 weeks would start counting letters on the standard visual acuity chart. By 1 month, she was counting 5 letters on that chart, and it's now been out over 2 years, and she's continued to see improvement when, in fact, this should be a progressive disease, and there should be an increasing loss in visual acuity. Since that time, we've treated another additional 28 patients. We haven't seen any major safety concerns related to the stem cell treatment. There are clear signs of engraftment and survival. So this is the implantation site. at 1 day. At 2 months, you can see the appearance of these pigmented plaques, and they continue to increase with the field days to 6 months. So during the 1-year follow-up period, the patients both in Stargardt's and in the AMD trial have shown significant improvements in visual acuity in the RPE treated eye, and the fellow eye, the untreated eye, remains unchanged, or they continue to show decline in visual acuity during that same time period. We also have a number of other programs that we're hoping to move into the clinic as well. We have a program in corneal repair generating corneal endothelium from embryonic stem cells. Corneal disease, actually 10 million people who have corneal blindness. It's the most transplanted organ. About a third of those are due to endothelial failure. And so the solutions involve either the transplantation of the entire thickness of the cornea, while more recently and more popular is DSAC, where you just transplant just the endothelium on desmase membrane. And we have now been able to generate entire sheets of these corneal endothelium from embryonic stem cells. They have been extensively characterized. We show the ZO1 staining for tight junctions. We can also show the sodium potassium ATPase pipe pump. We now have optimized cryopreservation so that we can ship these corneal endothelial cells worldwide. We've done a global gene analysis showing that the embryonic derived corneal endothelium is nearly identical to the adult-derived tissue. We've also been studying a number of other cell types in the eye field. These include the retinal neuroprogenitors, also photoreceptor progenitors, as well as ganglion progenitors. So with regard to the retinal neuroprogenitors, we've shown that these cells can reverse the progression of photoreceptor degeneration in these animals with retinal degeneration. When we look at the ERGs, when you look at the A wave, which is the electrical activity of the cones in the rods, we can see a very significant difference in their activity as opposed to the untreated animals. When you look at these animals histologically, what you are able to see is this is the outer nucleated layer. This is one dark line. It's pretty much gone, almost complete degeneration. By contrast, animals that we injected with these retinal neuroprogenitors, very significant rescue of the outer nucleated layer. Interestingly, when you do a PCR analysis of the retina, what you see is that you only have mouse cells. There are no human cells. So what's going on here? Turns out that when you inject your cells to the teal vein, for instance, most of them get lodged in the lungs or whatever. So it turns out these cells are acting distally in an endocrine fashion. So we've now started to look at those cells through antibody arrays and other methodologies to discern what's going on. So we can learn from these cells as to what the neuroprotective mechanism is. So we can see very clear differences between active and non-active cells. We have a program in hemangioblast research. So hemangioblasts are bipotential cells that have the ability to turn not only into the vasculature, but also into the lymphoid and myeloid lineages. So in this particular study, we labeled some of these cells with GFP. So again, we've been able to create very, very large numbers of these hemangioblast cells through our embryonic stem cells. And when you label them with the GFP and inject them systemically into mice that have ischemia, reperfusion injury, that if you apply pressure to the eye and it damages the retinal vasculature, you can see that cells within 24 to 48 hours start to migrate to the damaged retinal vasculature and incorporate into it. By contrast, the contralateral eye, untreated, there's no incorporation of these cells. So the cells are very smart. They know where to home from the damaged tissue. We've also looked at injections of these cells in mice that have ischemic limbs. So here's a Doppler flow behind top portion of the mouse. So there's no blood flow to this limb. One injection of the cells within a month, you can see complete restoration of the blood flow, very robust collateral circulation as a result. In a myocardial infarct mouse model, we were able to see a decline in the mortality of about 50%. This is the injection of these cells in some spontaneously diabetic rats. Again, as you know, all the horrible secondary complications of diabetes are mostly microvascular in nature. And again, you see very soon, within a few days, the incorporation of these hemangioblasts into the damaged retinal vasculature. But surely, obviously, these cells would probably be incorporated into other areas of the body that mentioned, these cells have high potential, so in addition to making the vasculature and taking LDL, if you change the culture media, you can turn them into lymphoid, myeloid, and other hematopoietic lineages. So we've been able to turn these into entire tubes of red blood cells that can transport oxygen just like normal transfusible blood. We've also gotten those cells to enucleate. As you know, our red blood cells are the mononuclei. We also have turned these into megakaryocytes and platelets. So the megakaryocytes are these giant multinucleated cells that start to produce these proplatelets, and then those are turned into platelets. So we've been able to now generate very large numbers and highly pure populations of platelets, both from embryonic stem cells, but also from iPS cells. And we've shown that both the iPS and ES-derived platelets are able to be involved with clot formation and retraction. They also incorporate in vivo into a mouse developing thrombus after laser-induced arteriolar injury. And we've also tested these platelets to show that they circulate in rodents that have had myelodepletion. When you look at these using EEM, you can look at the morphology and ultrastructure, and again, platelets are biconcave. So if you look at the cross-section, this is a normal platelet, and this is an iPS platelet, and you can see the characteristic alpha granules, the dense tubular system, the glycogen granules, and the appropriate organelles. So they're virtually identical to most of the tests that we were able to carry out. So we're in the process now, because you obviously need very large numbers of these cells for therapy, scale up using microfluidics and bioreactor systems. We're completing our pre-clinical testing for biodistribution, safety, tumorigenicity, that sort of thing. We've had our pre-IND meeting with the FDA for using the iPS cells for this application, and hopefully we'll be able to file an IND downstream. We also have a program using mesenchymal stem cells that we've derived from both ES and iPS cells. As I'm sure you're all aware, MSCs are found in the bone marrow, adipose, umbilical cord, even in tooth buds. They differentiate into fat, bone, and cartilage. They migrate to the site of injury, and interestingly, in particular, for clinical translation, they're immune privileged, so you can transplant allogeneic cells off the shelf. There are over 200 clinical trials listed in the U.S. under clinicaltrials.gov for a wide range of applications, but all of these applications are using bone marrow or other primary sources of MSCs. So why use pluripotent stem cell-derived MSCs versus adult primary tissue? First, of course, is that you can use these off the shelf. You can create very, very large numbers of these cells for immediate use. You can create an unlimited cell source. Again, these cells, you're starting out with a master bank where those cells are, again, immortal. They're very easy to derive. They can be expanded in very large numbers. In fact, we've been able to show that we get 30,000 times more units from embryonic stem cell-derived MSCs than you can get from an equivalent unit of adult bone marrow. So we can get very, very large numbers of these. But it's not just that you can get large numbers. If you expand adult tissue, for instance, you start to lose their biological potency. So actually, expanded MSCs are often a great negative controls. But with our cells, we can go through much greater expansion without the loss of that activity. So this just gives you an illustration of the efficacy of these cells in some animal models. In this case, we were studying multiple sclerosis in the EAE model. And in these untreated animals, the clinical score is 2 to 4, which basically means that they're partially to completely paralyzed in their limbs. By contrast, a single injection of our ES-derived MSCs, we were able to reduce the score below 1. There was actually no leg paralysis. And interestingly, we also tested in this model bone marrow-derived MSCs as a control, and we saw that there was minimal impact, actually pretty variable from those lots. The animals still showed significant paralysis. So in summary, we were able to show a dramatic reduction in the clinical symptoms of EAE. This was both prophylactic and therapeutic inhibition. We were able to show that these ES-MSCs had inhibition of the T-cell function. We've also discerned differential cytokine expression between the ES-derived MSCs and the adult MSCs. We've also seen differential ability to migrate into the damaged tissues, into the CNS. And so as a result of these data, we think that this is a leading candidate for clinical translation. We've also begun studies looking at these MSCs in lupus and lupus nephritis. Systemic lupus erythematous, or lupus, is an autoimmune disease that affects virtually every organ and system of the body. There's no cure for lupus. It affects the heart, joints, lungs, blood vessels, kidneys, liver, nervous system. And in 50% of those patients, they will develop lupus nephritis. That is due to immune complex formation in the kidney glomeruli that leads to dysfunction and failure. What you're seeing here is the glomeruli of a normal kidney. By contrast, when you look at a lupus patient, you can see the immune complexes, and eventually that leads to kidney failure. So we've studied either single or multiple injections of these cells in what's known as the NZB mouse, which spontaneously develops lupus nephritis. It's the gold standard, and it's very similar to human lupus. And we've seen that these ES-derived MSCs have a dramatic effect on morbidity, including proteinuria, kidney function. They also protect against death. They show marked improvement in survival after injection of these cells. So again, this is another candidate that we're pursuing for the clinic as well. So I just mentioned two examples, but again, there are literally hundreds of different potential medical applications. Just the autoimmune diseases alone, there are over 100 of those, including multiple sclerosis, osteoarthritis, aplastic anemia, lupus. They can also be used to treat liver, kidney, and pulmonary diseases. And again, the reason these are ideal is because there's no need for the immunosuppression. They only persist transiently a few days, a few weeks at the very most, and you can irradiate them and still show function in these animal models. And so I want to thank the people who actually did all the work here, in particular Shizhang Liu, Erin Kimball, Irina Klemenskaya, as well as clinical collaborator Stephen Schwartz, and a number of other individuals. Thank you.
Thank you. Excellent talk, and for keeping up the time, we have time for questions. Yes.
What are your plans
Yeah, so we're currently, obviously we're doing our clinical trials in the UK.
Could you repeat the question?
Oh, yes. He wanted to know what our plans were for clinical trials in Europe, and I pointed out that we already have one clinical site here in Europe at Mooresfield in the UK, and we're now in the process of designing our phase 2, so we're hoping to expand to at least a couple more sites here.
Yes, I have a question too.
Yeah.
Regarding your technology deriving pluripotent cells, you use either ES cells or iPS cells. Would you a little bit comment on whether you see differences if you use iPS or ES cells, and what is the technology advantage of deriving ES cells of your company?
Yes. So it turns out, for instance, for platelets, obviously there are many different ways of reprogramming cells to create IPS, and they vary very considerably, whether it's the micro or the macro RNA, whether you're using episomal methodologies. And so we've found a couple of those methods that work almost identical to what we see with embryonic stem cells. Indeed, when we make platelets, we actually can get better and larger numbers of platelets using the IPS cells. For other models, we're fine-tuning that, and so we find with certain methodologies that we've got complete reprogramming that they're almost identical to the embryonic stem cells.
Can I ask whether in the patients that you're treating for eye disease,
Yes.
Are they immunosuppressed at all?
Yeah, so we're not really sure if it's necessary, but I know from animal studies we've done in the past with dogs that during the perioperative period, when the cells are acclimating, that's when you got most of your cell death. That's when you can get sensitization. Now, the eye is an immune-privileged site, and in theory, you shouldn't need immunosuppression, but these are very advanced stage patients, so we're not really sure. So what we have done is a transient course of immunosuppression. So we start the week before with tacrolimus and MMF, And then at 6 weeks, we discontinue the MMF and go another 6 weeks with tacrolimus and then discontinue that altogether.
So in the long term, the patients aren't on immunosuppressants?
Exactly, right. In our phase 2, we'll try to address that to find out whether or not we need one, both, or none of them at all.
Very interesting talk. So I have a question regarding the purity of your cell-based product. How did you make sure that you don't have any pluripotent cell in your product? So how did you separate them?
Okay, yes. So we're fortunate in that our RPE, you can go through many months of expansion and de-differentiation and redifferentiation, so that what we actually do in the end is actually, it would be almost impossible at that point to still have an undifferentiated cell. But we've created antibodies where you can do your staining for your final released product, and we can actually throw in, for instance, 5 cells into a lot of 8 million RPE. We can find them all, so we're able to actually identify down to 1 cell in over 1 million. In addition, the FDA did make us do spiking studies where we had to go in with huge numbers of undifferentiated cells in the site that we're going into, in this case, the subretinal space, and we were able to show that even if we were getting 6 orders of magnitude more than our level of assay, we still could not deter it.
Thank you.
And in terms of the purity, these are 99% plus, I'd like to say 100%, because by all the various methods, they appear to be completely pure.
Thanks.
Would you think there is a difference in the therapeutic outcome if you deliver the cells systemically or locally?
Yes.
I'm thinking especially about subretinal transplantation in the eye compared to IV injection.
Right. So yeah, so that's going to be really very, very dependent on a particular disease. So in the eye, of course, we want to deliver the RPE to the subretinal space. But what we have seen in other models, for instance, whether we're using the retinal neural progenitors or whatever, that you can do it either intravitreally or you can do it systemically. Certainly, if you're going to go in for osteoarthritis, you would probably, it might be preferable to do them locally into the joint. So it's certainly going to depend on the disease application. And it's not always very clear, when we go in for these road models, we go in IP, but these cells migrate. So depending on the particular cell type, you may need to deliver them locally, or if they're able to migrate, then you can probably do it.
So with regard to the effects of the mesenchymal stem cells in the MS model and the lupus model, can you speculate a little bit what you think the MSCs are doing and how they're improving? Do you think it's a similar mechanism? Are there different mechanisms?
Right.
And do you think there are maybe preparations you can derive from the MSCs, which don't have cells anymore, which may have similar effects?
Yeah, yes. So it actually does turn out, one is that we can irradiate these cells, so we can actually show that those cells are functioning even after they've been irradiated. So the hope is that perhaps that we can isolate some of those components to figure out what's going on, as we have with the retinal neural progenitors. In terms of the mechanism, we know that in these various autoimmune diseases that there's a hyperactive B and T cell population, so we believe that these cells are down-regulating that. We can clearly show in vitro, inhibition of the T cell function. But of course, there's a lot we still don't understand about these cells. So, for instance, we're learning just now very significant differences between the ES-derived MSCs in the bone marrow have to do with their ability to not just home, but to extravasate into the tissue itself. So there's still a lot in terms of mechanism we're trying to figure out.
Last question.
Hi, could I ask you a question about the evidence you have for the location of the RPE cells after transplantation, both in the animal models and in the patient? So in the animal models, were you able to label the human cells and show their localization? And then in the patients, I think you showed pigmentation spots, which usually you see when the neural retina is destroyed.
Yes, so clearly in all of our preclinical studies, we go in with anti-human mitochondrial or nucleated antibodies, and we can easily show, even at the end of these experiments, that you can see the human cells lining up in the mouse model layer very nicely. So we are able to find those cells very clearly. In the humans, obviously the advantage of going in the eye is you can look into the eye in real time and see what's going on. So we can use OCT, for instance, as well. And where we can actually see down at the cellular level, and we can actually see the appearance of those cells attached to Bruch's membrane.
Okay, well, thank you very much, Robert. I know Robert has to fly back fairly soon, so if anybody's got any more questions, then grab him as quickly as you can. Covered more or less every tissue you can imagine. There was a picture of heart right at the beginning, led to movement heart regeneration as well. And this is subject Lee Chan from the University of North Carolina.